As Joe said, my name is Katelyn Maeba. I currently go to University of Manitoba and I'm entering my 4th year in the genetics program. And this is my 2nd year working with Bill, so I'm continuing a lot of my work that I did last summer as well as research that he's done in the past, working with milk fortifiers. Okay, so the shape of my presentation will be as follows. It continues a lot from last summer and looking at breast milk and supplements that are added to them right in nurseries and hospitals and things like that. As well as that, we went further and looked at oxidative stress on how fortifiers are affecting cells in the gut, or gut cells in babies, and whether these protocols are actually inducing osmotic stress. Bill's looked in the past on a specific fortifier, HMF, and so we also wanted to look at other fortifiers and combinations and see whether they induce oxidative stress. And then as well, Bill was also working in collaboration with Dr. Joe Corden, looking at a specific gene, BNIP3, and he believes that overexpression of this gene is causing cell death, so we wanted to look at that in my cell lines. And if they are inducing cell death, we want to figure out what kind of cell death, sort of what pathways are involved, and whether we can rescue these cells. All right, so human milk fortifiers are added to provide extra calories for growth and development of preterm infants. And our focus was on, of course, HMF, was one that Bill looks at specifically, as well as multi-component fortifiers and supplements that provide high levels of protein. So some of the new ones on the market are Beta-protein, there's Enfacare, Enfamil, quite a few different ones that we were working with. And we actually went to a couple of the NICU wards, we went to St. Boniface as well as Health Science Center, just to figure out the current combinations and brands that they're using right in the nurseries in the hospital, just so that we could work with those so it's current data that's used in Manitoba hospitals. So one of the first things we did was we wanted to measure osmolarity of some of the different combinations of fortifiers. Because high osmolarity has been implicated as a cause of necrotizing enterocolitis, so it's kind of an important issue. So as expected, osmolarity did increase with supplementations. This one I have highlighted here with the blue star, that is just the osmolarity of regular breast milk, and then all of these other combinations, they're combinations of fortifiers added to breast milk that are used right in nurseries. So the Enfacare A+ yielded the highest osmolarity, which is this one right over here, followed by this one right here, which that is the most recent combination of fortifiers that they're using in the nurseries. So that is a combination of Enfacare A+, premature Enfamil 24A+, and HMF. So again, these are all added to breast milk, and then I just measured them with an osmometer. So all of the combinations, not including the control, did have considerably higher osmolarity, and we definitely need to further investigate this issue, but of course time constraints, we haven't quite gotten there yet. So that identified our first problem with fortifications. And now, this is the second problem actually that my supervisor Bill identified a couple years ago with HMF. And that is that babies receiving more than 50% of their feeds with HMF, which is highlighted on the side here, have significantly higher urinary F2 isoprostanes, which are robust markers for oxidative stress. And oxidative stress is of clinical concern because there are many disorders of premature diseases linked to it. As I mentioned before, necrotizing enterocolitis is one of them. ROP, bronchopulmonary dysplasia, they're all directly linked to oxidative stress. So Bill had discovered this with HMF, and a natural question is why are HMF supplemented feeds associated with higher oxidative stress levels? So part of my project was to not only work with HMF, but some of these newer fortifiers and supplements that are being added. So what we did was to place these different combinations and supplements into breast milk according to the standard nursery and manufacturing protocols, and then we subjected them to a simulated digestion. So some of the processes was to include a gastric stomach phase, intestinal phases where we added different enzymes, changed pHs. We even had simulated kind of a stomach environment, the churning of the stomach. And this protocol was validated and published in Bill's lab a few years back. Okay. So after we had completed a fair number of these digests, we applied the treatments to enterocytes, the certain cell lines that I was working with, and we performed a couple DCF assays, which these are assays that measure the oxidative stress and reactive oxygen species in the cells. And so how DCF assays work is the DCF dye is applied to the cells after the treatments are applied, and the DCF dye reacts with different ROS, such as like hydrogen peroxide and stuff, so the level, and then when they react, they fluoresce. So the level of fluorescence is directly correlated to the level of ROS within the cells. So this graph right here is showing the results from one of the assays. So higher levels of emission correlate to higher levels of intracellular ROS. So what we did find is that these fortifiers are increasing the oxidative stress. The highest ROS production was found in this combination, which is again the combination that's currently used in St. Boniface NICU that had that N for care A+, premature infantile 24A+, and the HMF. Okay, so now this graph is again going back to some of Bill's previous research, that it's a quantitative real-time PCR microarray experiment in which the cells, which they were again the enterocytes that I've been working with, they were treated either with digested breast milk or digested breast milk that contained HMF. And so I just want you to note the fold increase in BNIP3. I have it highlighted at the bottom there. BNIP3 is part of the BCL2 family and induces cell death. And so this is kind of where my summer project began to collaborate with some of Joe Gordon's work, because he is very interested in this particular gene. All of the other upregulated genes are antioxidant enzymes, so you might notice the one MT3 here with the huge increase. It's an antioxidant that's involved in zinc transport. Didn't focus on that too much. So going back a little, just to kind of get you guys seeing where I'm going with this, Bill does a lot of work with human milk fortifiers, and so we've discovered that they increase intracellular oxidative stress, and then in turn this increase in oxidative stress is increasing the expression of BNIP3. So to determine whether this overexpression of this gene is inducing cell death, I performed several experiments using a couple different cell lines. The one that's left for you. The one on the left is a cell line known as KCO2 BBE cells, and the one on the right is HT29 cells. And they're both adult colon cancer cell lines that are brush border expressing, and they form confluent polarized epithelium cells that resemble colonocytes or enterocytes, or those of you who don't understand that, it's basically baby gut intestinal cells. These cell lines both take about 2 to 3 weeks to proliferate, differentiate, start maturing and developing tight junctions. So before I could start experiments, it would take me about 2 to 3 weeks of culturing, so that took quite a bit of time. After we got these growing, Joe's lab worked a lot doing transfection this summer. So my particular transfection, we used an affecting reagent kit, and we were performing transfections to overexpress BNIP3 and see just the results of that, taking a ratio of live and dead cells. So briefly, a transfection is basically a non-viral method for deliberately introducing DNA or genes into cells. And we chose to use the affecting transfection reagent just because it's a non-liposomal lipid formulation that had a high transfection efficiency. And we actually couldn't find any data that either of my cell lines had been transfected in the past. So we wanted to use something that had a high efficiency so we would be able to transfect them and then be able to look at the expression of BNIP3. So basically with the transfections, we had between 3 to 4 conditions most times using, we'd always have the control, which was an empty vector DNA condition, and then we had another plasmid containing BNIV3, that one was used to show the overexpression of BNIV3. So we would mix, basically you take the affecting hits, mix it with the DNA, and then you would apply those to the cells, and after about a 48-hour incubation period, we would stain the cells for imaging to determine the live to dead ratio. So the live dead stain was used to analyze gene expression and utilize 2 fluorescent dyes. So one of them was calcium, which stained the living cells green, and the other one was red ethidium homodimer, which stained the dead cells red. So after we applied the stains, we looked at the cells using the epifluorescence microscope. So you can see these are a couple images from one of the experiments I did on my CACO2 cells. And as expected, overexpression resulted in increased cell death. So this one was just the control that used an empty vector. And then over here, this had an overexpression of BNIV3. So you can see the ratio of dead cells to living cells. There is quite a bit more dead when BNIV3 was overexpressed. But of course, there still is death in the control as the transaction process itself does induce death on the cells. And then the next slide just shows the other cell line, EHG29s. Again, we found the same results, that the control had a lot less death than when we overexpressed BNIV3. And I actually am working on another cell line doing these same transaction experiments. It's a fetal cell line, FHS cells, but due to time constraints, we haven't quite gotten that one finished up yet. And then, okay, so after we performed some of these transactions and determined that BNIV3 is actually inducing cell death, we did do a couple rescue experiments in which we applied drug treatments in between the incubation, between the transactions and imaging, that we expected them to actually rescue the cells and inhibit cell death, but these didn't work quite as expected, and we actually did find more cell death in some cases. The drugs we applied were cyclosporin A, necrosat-1, and ZVAD. So we're not quite sure why they didn't work as expected. We're thinking there might be other cell death pathways that were activated. So in the future, we are hoping to do maybe some calcium imaging just to see what other pathways might be activated and why we're seeing more cell death rather than preventing cell death. Okay, so where does this leave us? After my transaction experiments, we can successfully transfect gut cells now, which that was one thing that we weren't even sure was possible. So this leaves us the opportunity to investigate cell death pathways, and it also indicates that upregulation of BNIV3 can have adverse consequences. So some of the conclusions from all of my projects this summer. Common breast milk supplements do significantly increase the osmolarity prior to digestion, and this was in addition to HMF that we had discovered a couple years back. So these may cause gastrointestinal distress and lead to other preterm disorders. We also know that HMF significantly increases oxidative stress, and we determined that other fortifier combinations are also increasing the oxidative stress, and this is consistent with other clinical findings. So going forward, we plan to exploit the overexpression model using BNIV3 and try to understand the mechanisms of this gene and BNIV3-mediated cell death. And then one more thing hopefully we're planning to do is identify components in these fortifiers which may be harmful, which are causing this oxidative stress, and possibly in the future designing better fortifiers. And I just want to give a special thanks, of course, to MICH for all your support in helping my research possible this summer. I need to thank all of the mothers for donating the breast milk. That obviously made my research possible. Of course, my supervisor, Bill Deal-Jones, unfortunately he's not here today. And then I also want to thank Joe Gordon and my massive lab that I worked with this summer.
Slide that the combination of formula they use in symbiotics gives this high response, right?
Yeah.
Did you compare it to the one that they use over here?
Yeah, we did. I had about 14 different combinations that we measured each of them on their own, and then we did combinations that they're using in the hospital. So we did measure all of those.
And so how is it so different between, say, Memphis and over here?
Some of them are similar, but some of them, I guess, they just use different combinations based on what the doctor is.
And do you know if there are they aware of differences in frequencies of certain diseases between the two centers? I'm not aware of it. You would expect if it is so important for some of these diseases that it would become.
Well, I think what we're trying to determine right now is more so which ones are sort of more toxic to the cells. But that, of course, we're in the process of discovering which ones are worse and which ones are better. So it's more up to the doctors right now what they think are better fortifiers and giving to patients.
It's one of the things we looked at in pediatric surgery with neonatology. Manitoba has almost half the incidence of necrotizing enterocolitis compared to the national.
Actually, I wasn't aware of that. So.
Yeah, so that is an interesting finding, we think, and this might be part of the information.
So along the same lines, I was just wondering if you knew whether or not, like if you look at all the babies born, because they don't all get NAC, but so if you look at prems born and you look at the ones that get NAC versus the ones that don't get NAC, has anyone ever looked to see if those babies are fed certain human milk fortifiers versus others, or maybe some don't get the human milk fortifiers?
Well, I know that's why Bill was really interested in HMF because that was one of the first ones that was linked to causing necrotizing enterocolitis, which is why they're starting to design new formulas. And then that's why we're trying to work with these different combinations to kind of see which ones are better and which ones are worse.
Okay, thank you, everybody.